Yeah, thanks, Johnny, and also thanks to Adrian for this excellent talk, which I think you will discover was quite kind of complementary on one hand to my talk, and also facilitating my talk, which is great. I will look at this partly in a slightly different angle, but you will discover that we were partly working on within the same project, but also have different backgrounds and perspectives. So I will first quickly summarize or review the scientific evidence for increased productivity in mixtures, something that was partly touched already by Adrian. And I will also go a little bit into this issue of yield stability, which already brings the time aspect into the whole story, which you, Adrian, didn't touch so much. But as I am also working with perennial crops with a willow system, the time aspect becomes really very obvious. And I think it should also be considered much more when talking about annual crops. And that's one of the questions that I will end up, as I already can say now here. And then the third issue I will briefly discuss is something that is already you, Adrian, mentioned, the diversity issue of diversity per se versus component identity. So how much does it matter which kind of species or varieties we use in a mixture? And that I will try to break this down a little bit into the properties or the characteristics of the mixture components and how they matter, and possibly show some ways how we could go more into this in the future research. And then I try to scale this up also more at an ecosystem level. I call this the ecosystem trait puzzle. So you have all these different, I call, when I'm talking about nutrient use efficiency, for example, you can break down, for example, the yield of a crop into yield components. And this could be when you are in a nitrogen limited world. It could be the capacity to accumulate nitrogen on the one hand, and then on the other hand, the capacity to make yield out of the nitrogen that is accumulated. That's something that I call for yield components in that context. And if you tease down these traits more and more, then you get a kind of a puzzle, and they can blow this up or scale this up on an ecosystem level, and then you get many components that all in the end contribute to the final yield. That's the story that I try to go through now. And if we start with the scientific evidence, that's a quite old slide from '99 that people have found that they started to look mainly in grassland systems and looked on average values. The doubling of plant diversity would increase biomass production of grasslands by approximately 20%. And that's, as I said, an old picture that was and that was confirmed also for other systems. And there are since then lots of confirmatory reports after that. This last one is from this year, based on average values. The grain yields in this case in intercrop systems were again about 20% greater than in the matched pure cultures, and the intercropping benefits, they increased over time. Here, the time aspect already comes in, and these authors already had some speculations on what could be behind this time effect, and they speculate on that it's through effects on soil fertility, but they didn't measure it. And then what is interesting is that in all these reports that are published, at least, they have, for statistical reasons and so on, obviously compared matched pure cultures with the corresponding mixtures. But what about if there is one single species or variety that maybe outperforms all the other mixtures? And this is something that we don't like in statistics because it's not kind of balanced. But for the farmer, he wouldn't care. The farmer simply would say, well, if I find one single species or variety that is better than all the others, fine. I simply take that one, and I don't care about the rest. And this is, of course, not the way we can look at it with our statistical eyes or academic eyes. And this is also shown in this. These authors have published this paper a few years ago, where they have shown that the maximum growth rate of specific monocultures and mixtures is often higher than average diversity effect on productivity. And that was confirmed for both grassland and forest ecosystems. And interestingly enough, this paper is not very much cited in the literature, which is also gives an interesting view. And but what it, in my opinion, also shows is that, something that Adrian, you already pointed out, that the species variety identity probably matters quite a lot. And this both in pure cultures and in mixtures. And you, Adrian, mentioned the breeding aspect in the whole thing. The monocultures or our crops are, of course, bred for certain monoculture conditions. And that will, of course, have an effect also when you grow them in mixtures, but nobody knows about it and that's why it's so difficult for us to predict it. And now I would like to come to this time aspect. This is from a paper that I learned a lot from. It's also quite old already. It's from a perennial grassland where the authors found that these beneficial effects of increased diversity on productivity, they develop over time. If you look here at the different times, the different years in 2000, and the species richness down here, in 2000, there was no effect of species richness. But in 2001, so it increased the effect of species richness, but mostly in terms of that if you grow less diverse systems, then the yields are decreasing. And they are not as much decreasing when you are growing them in more diverse systems. And that's, in my opinion, a rather interesting observation. And also here, I just would like to mention one of our own experiments using willows, a perennial system. And after 3 years of growth, we didn't find any significant species or genotype diversity effect on productivity. And that was in these systems. In a willow system, it's a rather short time, only 3 years. And then one can, of course, also, because we have a time perspective in this figure here, one could speculate on what implications that might have for yield stability. And then I would like to put your attention on these last on this highly diverse system, and over the years, though they are not significantly different in these, and that so maximal yields and no diversity effect in the first year, as I already mentioned. And how would that, what implications would that have for yield stability over time in annual crops? And on this, regarding this yield stability issue, I would also like to show you an example from the same experiment that Adrian already mentioned, the Diversify project, where we had trials all over Europe, our own trial here in Sweden, and then a Danish trial in the Scottish one that Adrian already mentioned, in Germany, in Spain, and in Italy. we had to use partially overlapping cultivar sets. Of course, we couldn't use the same cultivars across all these sites. That wouldn't have worked. And we used standardized assessment protocols for the plant traits and the crop performance. And one of the results that we have summarized here from these assessments where we were looking into the yield stability over time. And it is already a tricky issue on how to really methodologically assess a yield stability. There are a few tricky issues in it, which I don't want to go into detail at this moment. But we assessed, in this case, we used 3 different stability indices, and basically all 3 of them showed the same result. So we feel that this is quite robust. And here we calculated the stability index as the mean expected year-to-year yield difference expected from the growing the crops in pure culture, divided by the mean observed year-to-year yield difference in the mixed crop. And if you do that this way, values larger than 1 would indicate more stable yield in mixtures. And as you can see in this figure, which is for pea barley and faba bean wheat mixtures, that we found values either around 1 or even much lower than 1 in most locations, and which means little evidence for more stable yields in mixtures than pure cultures of these annual crops. And I should also add that this is from 2 years, to 2017 and 18. And 2018 was a rather dry year, as maybe many of you might remember, and 17 was a more normal year. So we really picked out some extreme years. So we can't blame that there was no difference between the 2 years. It was in most of the sites. Even if it's in the Italian case, for example, it was 2018 was actually a rather wet year. So it's not the same all over Europe with these differences. But what is interesting is that there is one big exception where we really found a kind of an over-yielding effect, and this is the Danish site. And this is at the same time the site where we had the best growth conditions. So we had expected actually that in those sites where we have more poorer growth conditions, there we would see more evidence for a stable yield stabilizing effect. But what we saw here was more in the opposite direction. So if we plotted the stability index against the observed mean yield, we found a significant relationship between the two. So if simply the crop was growing more, it was more likely that we could see a stabilizing effect of mixtures. And that might be related to the simple fact, that's our speculation, that if crops are not growing much, there cannot be much interaction. But if they are growing much more, then it might be more likely that they actually also interact, and that you can see also the positive effects of any interaction. And then I would like to briefly go into this issue of the diversity versus component identity issue, which also Adrian mentioned already. And here I pick out simply a experiment, the trial that we did here in Uppsala in Sweden with pea barley and faba bean wheat mixtures. And here specifically the faba bean wheat mixture, 50-50 mixture with 2 treatments, no nitrogen and additional nitrogen. And it was grown, as I already mentioned, in 2017 and 2018. And in all cases, we usually used 2 varieties of legumes and 3 varieties of the corresponding cereal that were combined in all theoretically different possibilities. And that gave quite many different plots, just to give you an an idea of what these kind of experiments usually mean in terms of experimental work. And if I take one example that is in one of the papers published by James Ajal, who was a PhD student in that project, we see that in our site, the mixtures usually favored the cereal grain yields. So we can see here the cereal is disket, for example, grown here in pure culture, and then disket grown in mixture, and then boxer would be the legume grown as a pure culture and grown in mixture. And the same here with alderon and boxer again. And what we can see that in general, here in our conditions, the cereal grain yields were favored. I don't think, Adrian, you saw the same pattern in your site. I think it was the other way around, wasn't it? And the second issue that I would like to point out is that a similar one that Adrian already mentioned, that the effect of the mixture depended very much also on the component identities. So if you compare the pattern here in the left-hand panel and the right-hand panel, it looks rather different, if you put it like this in a simple way. So the mixture component and the variety of it, it matters, but we don't really know why. And that's where we started to go into what I initially called the yield components in terms of nitrogen use efficiency. So we split it up into the nitrogen uptake efficiency, partly responsible for forming a yield, and the second, how efficient is the plant to make yield out of the available in the plant nitrogen, which is often called the nitrogen utilization efficiency. And if we then again look into these combinations, then we can see that here the pure cultures are in blue and the mixtures are in gray. So we usually, in general, we get that mixing result in higher nitrogen uptake efficiency, at least in some of the combinations, but lower grain yield per unit plant nitrogen, which I called previously the utilization efficiency. So we get more nitrogen is taken up, but they are worse in making yield out of the nitrogen that I have in the plant. And that's really interesting. If you compare the different variety combinations, you see there are big differences between them. They don't do it all in the same way. They do it and in disket, for example, there's no difference between whether it is grown in mixture or in pure culture in both years. I should also say here, and also in terms of nitrogen uptake efficiency, there is a little difference, but it's also relatively small. So this is, in my opinion, a way where we can try to analyze and to a little bit better understand why certain combinations of varieties work nicely together, where we have some kind of complementarity and where we maybe don't get this kind of complementarity. This is one way to come closer to it. So basically, in other words, how does mixing different species of varieties affect the individual traits that would contribute to finally the grain yield that we are interested in? And that brings me to what I called the trait puzzle that I so far have tried to discuss a little bit at the within plant level, just looking on different traits. But of course, that trait puzzle would extend also beyond the single plant and even the plant community. And to illustrate some of those examples, I will just to show in between this conceptual scheme here, what we have been talking about so far is how plant traits might affect the crop growth and then, for example, biomass or grain productivity or whatever. But of course, there are lots of arrows and connections also between single plant traits and how they affect pest and disease resistance or attractiveness, and how they would affect microbes in the soil. And those would again affect things like resource availability, resource cycling, soil carbon accumulation, and also quality issues. And they are, of course, also very important. And that's the trait puzzle that I'm trying to go into. And for that, I would like to use an example from a perennial system. Our EcoLink Salix field trials that originally were established in 3 different sites in Uppsala, in northern Germany in Rostock, and in Freiburg in southern Germany. It was established in 2014, where we had 2 to 4 different species of varieties that were grown in pure cultures and in all theoretically possible mixtures. And we have 3 replications per site, and further details are in this paper by Stefanie Höber, who was also a PhD student here. And if we just start to look from a crop perspective, of course, that's nothing new. We find significant differences between different varieties. That's not, as I said, that's nothing new. But it was more interesting than to ask the question, okay, if we then grow these different varieties, or that partly even are different species, if we grow them in a mixture, would the result be simply the sum, if we grow them in a 50-50 mixture, simply the sum of the 2 components that are grown in it? Or would we get a kind of a surplus, what people call an over-yielding? And in this case, as you also, it's illustrated in this figure here, where we have the observed growth or shoot biomass growth in the mixture against the expected from the pure cultures. And you see they are more or less falling on a line, a 1-to-1 line. So 1 + 1 is basically 2. Bit boring. The admixing effects on stand biomass were explained basically by the summed effects of the individual varieties assessed in pure cultures, no over-yielding. That's when we look on the final product we are interested, the shoot biomass growth. But again, if we now try to go a little bit deeper into the details and look into these yield components in terms of nitrogen use efficiency, here I now show only the result for the nitrogen utilization efficiency, which is the shoot biomass per nitrogen in the plant. And then we suddenly start to see that at least for one of the varieties, what we observe in the mixture is actually more than what we would expect from only the monocultures that are included in it. So there seems to start something to happen here after these 3 years. And we are now, of course, very curious to see whether this finally also develops to a difference, to a mixture effect in terms of yield in the end, which we don't know yet. But maybe there is already an indication of that something is going on, but it takes quite some time to really develop. And if we now go a little bit more into towards the ecosystem perspective, litter decomposition is something that in a perennial system is rather important for the whole nutrient cycling system. So we looked also here into the litter decomposition between and compared it between the single varieties included here that happen to be also different species, Luden on one hand and Tura, and again rather boring kind of outcome. The litter decomposition of the mixture was exactly in the middle, intermediate between the 2 original pure cultures, which again means that we have a situation with 1 + 1 is more or less 2, which means the admixing effects on decomposition were largely explained by the summed effects of the individual varieties. And increasing diversity did not affect decomposition. And then we looked also into the effects of these different mixtures on the mobilization or availability of particular nutrients in the soil, because my colleague in Rostock, they have a particular focus on and particular interest in phosphorus mobilization. And they look into the root-associated fungi, a certain group that is called dark septate endophytes that are responsible for or that contribute with an acid phosphatase activity, which is important for phosphorus mobilization in the soil. And there we found strong indications that when we grow Luden in mixture compared to pure culture, this phosphatase activity is much higher, and the same appears also for Tura. So that means that if we would put it in this kind of simple way, 1 + 1 would actually be more than 2. And the admixing effects on soil enzyme activities, they were clearly larger than the summed effects of the individual varieties. Increasing diversity enhanced the enzyme activity, which of course in an ecosystem perspective can be really relevant in the longer term, especially when it comes to phosphorus, which can't be taken anywhere else from. And that's basically the story that I wanted to tell here. And now I have only some final reflections left. What I already mentioned in the beginning, time seems to be important for mixture performance compared to pure cultures. And the question is then, to me, how can we make best use of the time effect when we are talking about annual cultures, where we sometimes have different histories, so to say, and which we often don't have control of, but they probably are important in the outcome. And the properties of the mixture components, they are important for mixture performance compared to pure cultures, and we need to learn more about which specific properties they are and how we can make use of this knowledge, for example, in breeding for mixtures. And then even if mixtures don't have, or we can't see any positive effects of mixtures on productivity compared to pure cultures, they can have positive effects on other ecosystem processes. For example, soil nutrient mobilization that we can't see at the moment, but that can be important somewhere in the future, again, bringing us back into the time aspect. And the final reflection, this complicated crop and ecosystem trait puzzle that makes predictions of this mixture performance really challenging. And then the kind of a question that I'm asking myself is, instead of really trying to figure out what is behind these differences, why some of the combinations simply work and others don't, instead of really trying to figure out why this is, it's more successful and a quicker way to just a kind of a trial and error approach, where we use local knowledge, which is there, as we have learned in the Diversify project. And we use the local knowledge and the local varieties to find the best plant teams. And then maybe we from the academic side try to find out why this is the case. And if we go again to this time aspect, maybe we face a situation like it is shown on this figure here, that we, in the short-term perspective, we might be more successful with monocultures or homogeneous teams, as it is here called, and that yeah, the most benefits we really can take out in a more long time perspective, which is, however, more difficult to plan. But what is a bit funny is that this figure is actually not taken from a plant paper. It is taken from a business leadership company, and it describes how human people work together. And I'm wondering still sometimes whether we don't see a similar pattern also when we look into plant teams, that we simply have to wait for several years to really make best use of more diverse teams. And that's my story so far. Thank you.